All right, so for our next section, we're going to start off looking at, again, root pressure. If you look at a plant, especially on a really warm, humid morning is usually when you can see this the most, transpiration from the leaves is going to be nil, basically. It's going to be almost none, if none at all. Remember, transpiration is evaporation of water from the leaves, so if it's really hot and really humid, especially, there's not going to be as much of a push for water to evaporate from the leaves. So in that case, that's a situation, very specific conditions, when you can actually observe the effects of root pressure. And so what will actually happen here is that water is going to be pushing into the roots. Remember we talked about in our previous section, we talked about how ions are going to move into the roots because of a proton pump, and as water moves into the roots, water is going to move into the roots as well. So in effect, water is basically pushing its way into the roots, trying to follow these solutes that are continuously being pumped into the roots. And so if that water pressure pushing in is the only pressure that you can see, if there's no water evaporating from the leaves, what will eventually occur here is a phenomenon known as guttation. And so this is where water itself will actually come out from the leaves in droplets. You can actually see droplets of water sitting on or especially under or on the edges of a leaf. It's not dew. It's not what you find in the morning, when you go out and the grass is wet. It's not that kind of water. This is water that's actually moved from the soil into the roots, up the stem, out the leaves in a completely liquid form. And so you can actually see water as it exits the stomata as water as opposed to water vapor, liquid water as opposed to water vapor. So that root pressure can be in some cases the only pressure present, and that's when you can actually observe it. But most of the time, when water is moving its way up the xylem throughout the rest of the plant, we're talking about transpiration. Right? That evaporation of water from the leaves is the primary force pulling water up the plant and eventually out from the leaves into the air. And so this water potential we talked about before, water potential has to be higher in the soil than it is in the roots, because remember, water always goes to the smaller and smaller and smaller water potential side of the equation. So if the water potential is higher in the soil than it is in the roots, then water is going to tend to move into the roots. And it's going to, from there, move its way on up the plant. And so as we move up the plant from the roots, the root hairs, into the larger roots, into the stem, into the top parts of the tree, into the leaves, and eventually into the air, we're moving in a direction that's more and more and more negative in terms of water potential. Right? The further you move up the plant, the more negative water potential you see, and that's of course why water moves up the plant in the first place. So that's the reason why water is moving. The effects of this transpiration, the force we're talking about here, is fairly substantial, and so you can imagine if you try to pull water up over a really, really long distance, 100 or 200 or even 300 feet, the amount of force you're pulling with is fairly strong, and so what could actually occur is you could actually break the water column. If you pull the water molecules too hard against the force of gravity, you'll actually separate those water molecules from each other, and they're going to fall back down the stem. Now, fortunately, there are a couple of reinforcing factors that I talked about in our previous section, cohesion and adhesion. And so since the water molecules are sticking to each other, and since the water molecules are also sticking to the sides of the xylem tubes, they are reinforced and therefore less likely to come apart from each other. We can look at the tensile strength of water. How much force it takes to pull this water column apart from itself is an inverse function of its relationship with diameter. So the larger the xylem tube, the less tensile strength there is. The easier it would be to break the water column. The narrower the xylem tube, the harder it's going to be to break that water column, because you're going to have more associations of water, not only with itself, but also with the walls of that xylem tube. So these xylem tubes are dealing with tracheids in all vascular plants, and then vessels in just the angiosperms, are very, very small in diameter to help take advantage of this relationship. The narrower they are, the stronger the water column is going to be. The stronger cohesive force you're going to see, and the more adhesive force you're going to see as well, if that tube is very, very, very thin. One thing that can occur, and this does occur actually frequently in plants, is a function called cavitation. Cavitation is where you start introducing air bubbles into the water column. And air bubbles are not a good thing as far as the plant is concerned, because what they can do is they will eventually block a xylem tube from functioning. So we see over here in this picture, we have an air bubble that's formed in this xylem tube. It's traveled its way up the tube until eventually it hits this plate. And once it hits that plate, it's going to serve as a blockage. It's like a roadblock. So these water molecules, in some cases, can bypass it and continue moving up another xylem tube. But in some cases, unless you start putting in new xylem tubes, then you're going to greatly limit the amount of water you can actually move through the plant. And that's the reason why plants do secondary growth, not only for strength, but they also do it to produce more xylem tubes to replace the ones that have been lost to cavitation. Um, there are several different adaptations that have occurred in different plants to help avoid this or minimize the damage caused by cavitation. There could be alternate pathways like we see in this picture, so we're going to bypass the blockage and then move into the adjacent xylem 2 over here on this side. There can be differences in pore sizes. For example, these pores here could come in different sizes to help minimize the effects of the air bubble. There could even be, in some plants, they actually have the ability to withdraw oxygen from this air bubble, thereby decreasing its size and allowing water to go back through the pores one more time. So that's another possibility too in some plants. Tracheids and vessels are involved, of course, not only for water transport, they're also involved in mineral transport. These minerals coming in from the roots, of course, are going to eventually get to the other, especially metabolically active parts of the plant where the nutrients are required. So primarily here we're dealing with phosphorus, potassium, and nitrogen. And so if you buy fertilizer off the shelf, you buy fertilizer, typically those are the three main ingredients of that fertilizer. Phosphorus, potassium, and nitrogen in some proportion is going to be what you buy off the shelf at a greenhouse or at a garden store, that kind of place. Iron can be in there as well. Sometimes they'll come with iron because iron is important for certain aspects of the plant's biology as well. But they're going to come in also with the water in the roots and be transported throughout the plant. Calcium is one that's actually kind of funny. It doesn't get translocated real well. Once it gets deposited, it doesn't really go anywhere after that point in time. The nitrogen, for example, can be translocated. It can be moved around from one place to another. Calcium, though, is pretty recalcitrant. Once it gets to where it's going, it's not going to come back out again. So we can look also at transpiration and take a look at what our guard cells are going to do in response to evaporation. If we look at the water taken in from the roots and where it eventually goes, the vast majority of it is going to go into evaporation. Most of the water the plant takes in never really gets used for much of anything except evaporation. Now, I mean, obviously the other 10% of the water is what's actually being used, and that's the really important part. I mean, we're losing 90% to get at the 10% that we are keeping, so we can use that. And one of the places the plants use the 10% is in photosynthesis, right? So water serves as an electron donor to help fuel part of the photosynthetic process, which is incredibly important not only for plants but for the rest of the living world as well, animals and fungi and everything else. The other aspect here that we have to think about is that photosynthesis also requires carbon dioxide. And so there has to be a way for water to get out of the plant as well as oxygen to get out of the plant, and we have to have a way for CO2 to get into the plant. And so under really hot, really dry conditions, stomata, openings on the underside usually of the leaves, can be opened or closed to help control water loss in the short term. Over longer term periods periods of time, it's up to other processes like development of C4 pathways, for example, to make waste during photosynthesis less important under hot, dry conditions. There are other more complicated pathways to help minimize water loss as well. But what we're seeing here is that the stomata, when they're open, I mean, that's great. You can bring oxygen out, you can bring CO2 in, that's great. But the drawback to it is if stomata are closed, then you're not going to be able to do that gas exchange. And stomata are open, water loss can be huge, which is not always a good thing if water is in short supply. So if a plant starts to become dehydrated, it can close the stomata. But then again, once you close the stomata, then breathing becomes more difficult, and you start running into problems with photorespiration and some other things that can come into play once stomata are closed for the long term. So the guard cells that are involved in opening or closing the stomata, we'll get back to this last picture for a second. Imagine a pair of balloons, like long skinny balloons that you tie together the ends, and in the middle here there's a little bit of a gap. And so these two balloons represent guard cells, a pair of guard cells here, and we have an opening in the middle. Right? Now imagine what would happen if you took those guard cells, or those balloons, and wrapped duct tape around them in this manner here. Duct tape wrapped this way and that way and this way and this way and this way and that way. So because we have relatively more duct tape on the inner part of this curve than the outer part of this curve because of how we wrap the duct tape, if you were to inflate these balloons more, what's going to happen is they're going to bulge outward on the top and bulge outward on the bottom, thereby opening up the stomata to allow for gas exchange to occur. Now, in real life, in a plant cell, obviously we're not dealing with duct tape, but we are dealing with something called cellulose, which is the material we find in the cell walls of plants. We find that cell wall deposits of cellulose are asymmetrical. They're uneven. And so on the inner part of the cell, like on this cell right here, there would be more cellulose on the inner part here and the inner part here, so that when this thing opens up, it actually allows for an opening into the cell to the mesophyll so gas exchange can occur. The stomata are going to open up under conditions that are not relatively hot, relatively dry. That allows for water loss and oxygen loss. It also allows for CO2 entry. When it's time to open up the guard cells, what's going to happen here is that there's going to be active transport of three different things, 2 ions and 1 sugar into these guard cells. There's going to be active transport of potassium ions, active transport of chloride ions, and active transport of malate, which is a sugar, into the guard cells. So what we're doing is we're spending a bunch of energy to fill up the guard cells with ions and sugar. Think about what water wants to do. If all of a sudden these guard cells have a really high concentration of solute, where does water want to go? Water obviously wants to go into the guard cells too. Water's going to follow. And so when water rushes into these guard cells, they're going to bow outwards on the top and bow outwards on the bottom, and that's going to allow the stomata to open up. When it's time for the stomata to close, what's going to happen is that sucrose now, different sugar, but sucrose is going to be pumped out of the guard cells, usually at night. And when that sugar is pumped out of the guard cells, so we're removing sugar here, when water is pumped out of the guard cells, when sucrose is pumped out of the guard cells, water is going to follow along behind it. So water is always going to follow the solute. If you move sucrose out, water is going to follow behind. If you move potassium and malate and chloride in, water is going to follow in behind. So water always goes where the solute goes. Transpiration can increase especially under conditions that are especially hot and especially dry and that are especially windy, because all of those things help to increase evaporation. The types of processes that regulate stomatal opening and closing often involve abscisic acid, which is a plant hormone, one of the more important plant hormones. And what this does is it allows for stomata to close during the day if it's an especially hot or dry day. Because, again, what happens here under hot, dry days, evaporation levels are high, water loss is high, and that can be very bad for the long-term wellness of the plant. So it's going to close the stomata to help conserve water in the short term. Now, again, that can only happen for so long because with the stomata closed, it's hard to bring in CO2, it's hard to get rid of oxygen, and that creates other problems down the road for the plant. But in the short term, this is a good way to help prevent too much water loss on an especially hot or especially dry day. All right, so one of the patterns too that we see with regulating stomata is that, especially under conditions where you have a lot of blue light coming in, blue light coming in often stimulates the initial movement of potassium ions into the guard cells, which then triggers the chlorides and malates to follow behind along with the water. When CO2 concentrations are high, and another trigger to close the stomata actually is if the plant has plenty of CO2. If it has enough CO2 to do photosynthesis, you might as well go ahead and close the stomata to conserve water, use the carbon dioxide you have, and then you can open up the stomata later on. Right? It's better off to close the stomata if you have enough CO2 so that you don't lose water that you don't need to lose. Right? It's wasteful to do that. So if you have enough CO2, close the stomata up, maintain your water, use the CO2 you have, and then maybe open them up later in the day to allow for more gas exchange to occur. The opening, like I mentioned, typically corresponds to blue wavelengths of light, which are some of the especially in the morning, plants are going to be getting more blue light than anything else, so it makes sense that would be a good trigger to cause them to open up their stomata and begin the process of gas exchange. If the temperature gets to be too warm, or if the air is too dry, then stomata are typically going to close, typically between about 30 and 34 degrees Celsius. That's going to be a point in time when stomata are going to begin to close. And especially for plants that have alternative mechanisms like the CAM pathway or a C4 pathway to do photosynthesis, when conditions are this warm for extended periods of time, these other plants, the CAM plants and the C4 plants, are going to have a very much decided advantage when it comes to maintaining water, but still being able to do photosynthesis. There are all kinds of other stress responses to water. So if water is in too large of a supply or not enough supply, we start to see a lot of adaptations to help to cope with that. We can see plants that go dormant in the drought season, so we're not so used to thinking about this in this part of the world. Usually our plants lose their leaves in response to cold and shorter days, but in the tropics, where winter is not really the same thing that it is here, those plants respond to the dry season by dropping their leaves. They have what we actually call drought deciduous plants, where in a dry season the plants lose their leaves, and then they regrow them in the following rainy season. So that allows them to maintain a favorable water balance as well. We can look at deciduous plants, like we mentioned, dropping leaves in the winter when water would actually freeze, and that would be a stressful situation as well. We can have leaves that are covered up with a very thick cuticle, which is going to help minimize evaporation even more. We can have leaves that are covered up with trichomes, and we talked before about how trichomes can minimize movement of air over the leaves, thereby creating a stable boundary layer of humid air, which helps to minimize transpiration too. We can look at plants that have fewer stomata. Right? So if they can get by having fewer stomata, what that does is it means you're going to have an overall less water loss than if you have more stomata. And this plant up here actually has what are called recessed stomata, where the openings are all the way tucked back up inside of these little chambers here, where there's going to be minimal airflow, and that's going to help minimize evaporation. So this plant here has got lots of stuff going for it. It's got a thick cuticle, it's got recessed stomata, it's got trichomes, it's got all kinds of things there to help minimize evaporation of water. The opposite end of the spectrum, flood conditions, can also be bad. Roots especially need to have oxygen, right? So they're going through cellular respiration. They need to have oxygen to fuel that, so they need to have an access to air effectively. So what can happen in plants that are typically in extended periods of flooding or high water is they're going to either have a lot of lenticels on the above ground parts of the plant. Remember, lenticels are for gas exchange, usually in a stem. They can also have adventitious roots that go from below the water out of the water to allow for gas exchange to occur there. We can look at plants that have what are called erenchyma. Right? So this plant here is a water lily or a lily pad. The top part of the leaf, there are tons and tons and tons of palisade cells for photosynthesis. And then down here, we have these great big old cells, air spaces and cells that are called erenchyma cells. And what that does is it allows the plant to float on the top of the water. They can collect their oxygen from the top parts of the leaf. So in this case, the stomata are going to be at the top of the leaf because this part down here is going to be underwater. It's not going to do you any good to get oxygen if this part of the leaf is below water. So stomata are on the top part of the leaf, and oxygen is going to be transported below the level of the water from there. Salt water is especially challenging because salt water creates not only the issue with flooding that we saw in the previous slide, but it also creates a very unfavorable osmotic environment where water would tend to be pulled out of the plant. There are several different mechanisms here. This mangrove tree in this picture is using what are called nematophores. All these little things coming up out of the sediment are the ends of roots from this plant, not in the entire picture, but right around this. This is from one plant. And what they're doing is collecting oxygen, setting it below the level of the sediment to the roots, and they're also going to help them control their salt balance. Succulent leaves are leaves that contain large amounts of water, typically very thick, very much full of water, and what that does is it helps to dilute the salt as the salt comes in. Those very watery leaves help to minimize the effects of that salt if the salt comes into the plant. Other plants can either secrete salt through what amounts to like a salt gland if it were an animal, or they can block salt uptake altogether. Some plants, though, are what we describe as being halophytes, and halophytes are plants that actually like salty conditions. The name halophyte means salt lover, and so these things can grow in places where other organisms couldn't. And the way that they do this is they actually create a favorable osmotic gradient in the roots. They create a bunch of carbohydrates, for example. They store those carbohydrates below ground, and so if the roots are in contact with a very, very salty environment, normally what would happen would be that water would exit the plant and go into the soil. But what these plants are doing is by creating this high concentration of molecules in the roots, they're basically making the roots isotonic to what's going on in the environment, if not hypertonic to what's going on in the environment. So water either will be balanced, or water will even go from the outside of the root to the inside of the root. Rather than being hypotonic, this high concentration of solute inside the plant roots makes the plant either iso or hypertonic to the outside environment. Now, the last section we're going to look at here is phloem transport. We're dealing now with carbohydrates that are being produced in the leaves that are going to be sent to the rest of the plant. We call this translocation, and this again involves primarily sugars that can be involved in producing larger carbohydrates or other things throughout the body of the plant. The phloem can also be involved in transporting hormones and mRNAs and various other substances as well. So not only that, sugars, amino acids, various other organic acids, proteins, ions, all kinds of things transported in the phloem. Remember, phloem cells are the ones that are alive, whereas xylem cells are not alive at maturity. Phloem cells are living because they have to be able to do some things that only living cells can do. So the idea about how phloem moves throughout the plant is known as the pressure flow hypothesis. This describes how carbohydrates move throughout the body of the plant. The way we learned about this actually was by using a type of insect called an aphid that is a sap-feeding insect. What they do is they take their mouth parts, their stylets, they insert them into a plant, and they tap into the plant's vascular system. They feed on carbohydrates, right? So what somebody figured out they could do many, many years ago was take an aphid, decapitated, as mean as that sounds, but then they could actually measure the rate of sap flow out of the disembodied head of that aphid. And they could actually get a rate and a volume of liquid estimated coming out of that aphid's head. So there's a long history of studying this in plants, trying to figure out how much sap they move, where they move it, the extent that they move it, when they move it throughout the year, and so forth. Generally throughout the plant, there are both carbon sources and carbon sinks. Carbon sources would be parts of the plant that are involved in producing carbohydrates, like the leaves, for example, produce glucose. Carbon sinks are where the carbon ends up. So a carbon sink would be like the wood of a tree, it would be flowers, it would be fruit, it would be any part of the plant that's involved in consuming carbohydrates. That would be a carbon sink. All right, so the way this carbohydrate gets into the phloem cells in the first place, right? The source for carbohydrates is in the leaves. And so what's going to happen here is these carbohydrates are going to be moved actively into the phloem cells. Phloem cells are going to do active transport from the mesophyll cells into the phloem. And since that's active transport, it's going to involve the use of ATP. I mentioned before, phloem cells have to be alive at maturity. When they're functional, they must be living for two reasons. They have to have a plasma membrane equipped with transport proteins, and they also have to be able to process or manufacture ATP. They're doing active transport, they have to have the pumps, they also have to have the fuel for the pumps. So they have to be alive at maturity. So what happens here is that once the carbohydrates are pumped into the phloem cells, water is going to follow in behind it. Water always follows the solute. So if we have carbohydrates now in the phloem tubes, water is going to go along right behind that carbohydrate and follow it. What that does is it generates a pressure up there in the leaves because all these carbohydrates, all these water molecules are pushing into the phloem tubes. That's going to help drive the phloem or drive the sap down from the leaves toward the roots of the plant. And so it's moving in this case from the top to the bottom due to pressure being initiated at the top of the plant because of the carbohydrates being moved in through active transport and also because of water moving in through osmosis. Eventually, once the carbohydrate gets to the sink, which could be the roots, it could be somewhere else in the plant as well, sucrose is removed, water follows behind it, and eventually that sucrose typically is going to get stored up in some way. If it's in the roots, the sucrose will be stored up as starch. If it's somewhere else, it'll be stored up as something else. But it's going to be removed from the phloem and stored in some form or fashion once it gets to where it's going. Right? So that's the end of our section on plant form and plant transport in particular. So we're going to cut this off here, and we will start a section on plant reproduction.